Good to go. Okay, well, good afternoon, everybody, and thanks for coming. Thanks for those of you who are joining us online. Thank you for being with us, albeit virtually. And today we have the distinct honor of hearing from Dr. Jane Carlton as part of our Dean's Lecture. And as you all know, the Dean's Lecture gives us an opportunity to hear from our newly appointed and promoted professors and senior scientists. And it's a great opportunity to hear about their work and getting to know them even better. So as I said, I'm thrilled to be able to, I'm thrilled that we're able to hear from Dr. Jane Carlton today. She's a leader in the field of comparative genomics and a true champion in our efforts to eradicate malaria. Jane joined Hopkins in 2023 as a Bloomberg Distinguished Professor and Director of our school's Malaria Research Institute. And as I'm sure all of you know, the Institute has been a research powerhouse all the way back to its founding in 2001, driving new discoveries and advancing prevention and control at field sites really worldwide. And Jane stepped into this role previously held by our Nobel laureate, Peter Agre, who just joined us. And truly, we could not ask for a better successor or a better leader for this very difficult moment. Each year, as I think most of you know in this room, malaria kills more than 600,000 people, most of them children. And yet, as we are all painfully aware, recent federal directives are disrupting public health work worldwide. At a time when we need to expand our efforts in malaria prevention and control, we are seeing programs shuttered and research canceled. Now more than ever before, we need to lean into innovation and strengthen our partnerships and not lose sight of our goals. The road ahead will not be easy, but there is still so much reason for hope. We have new vaccines. We have 45 countries that are malaria-free. And we have the great promise of omics and AI, which we'll learn about more today, which can supercharge our research. I know Jane is ready to seize on these opportunities and many, many more. As we look to meet the current challenges, we are grateful for the experience and passion that she brings to the school and the university. For the past 3 decades, Jane has been building a name in global health, using the power of comparative genomics to probe the biology and evolution of parasites that cause disease. And just this year, she was elected to the American Academy of Microbiology in recognition for her scientific contributions. Jane is an expert not only in malaria, as you'll find out, but also on trichomoniasis, a common and curable sexually transmitted infection. And like malaria, it's caused by a parasite. And in fact, I learned, she was on the cover of Science for sequencing the trichomonas genome, and she celebrated that event by ordering a beautiful cake decorated with parasites and gave out condoms and packages that were decorated with trichomonas. I'm sorry I missed that event, but it sounds interesting. Jane does groundbreaking research and clearly throws great parties, but her work goes much, much further than that. She is passionate about building collaborations between researchers and clinicians and translating discovery into real-world impact. She spent 20 years working in India, most recently as director of an NIH International Center of Excellence for Malaria Research. She continually worked on high-impact projects that brought together clinicians, basic scientists, social scientists, as well as field teams. Through her collaborative approach, Jane and her colleagues have advanced our understanding in epidemiology, transmission, and genomics, while bringing technology and training to the next generation of the malaria fighters. This is just one example of how Jane has built a career defined by cross-disciplinary approaches and, very importantly, the building of partnerships. Of course, she brings these same strengths to her work at the Bloomberg School. I know her colleagues appreciate not only her scientific rigor and her expertise, but her collaborative approach to leadership as she helps guide the Institute towards new innovations. We are especially excited that she is establishing a genomics and bioinformatics core facility, which is supporting and expanding our omics work. Jane will talk more about that today, and will also share some thoughts on both the history of the Institute and the field, and the current moment we're in. And as difficult as this moment is, and we're all feeling it, we know we cannot lose sight of our goals and our mission. And we have a strong foundation that will help us push forward. I am convinced of that. Our school will be making progress on surveillance, treatment, and control. We will also keep on breeding mosquitoes by the millions. That always makes me a little nervous, but we will keep on making discovery after discovery in the pursuit of eradication of this deadly disease. And while we get there, I'm sure Jane will order us a really fantastic cake when we reach that point. So Jane, we're so fortunate to have you here at the school and at the university, and we're just thrilled to be able to celebrate you and your appointment. You've been here for a couple of years now, but still, we're so delighted to have you join us today, and we're looking forward to your remarks. So the floor is all yours.
Well, thank you so much, Dean Ellen. That was a lovely introduction. And I'll be happy to provide you with some condoms afterwards if you would like. I actually still have some. Oh, the cake. Okay. I'm actually a little scratchy today with my voice, so I apologize if I keep taking sips of water. I'm going to be talking today, as Dean Ellen very eloquently described, all about malaria, history, current status, and then I'll be talking a little bit about my work and my lab's work that involves omics and machine learning. So, malaria history. I included a few slides about this because I wasn't entirely sure about the audience. Malaria is an ancient disease, as many of you may know. Along with HIV, AIDS, and TB, it's one of the top 3 infectious diseases worldwide. And it was really first described back 400 BC by Hippocrates, and then also by Neijing in an ancient Chinese medical text where they discussed the repeated fevers of malaria. Julius Caesar gave the first public health response to malaria through draining swamps around Rome. And then in the 1630s, the Jesuit missionaries came back from Peru with what was called Jesuit powder, or quinine, as we now know it to be, brought back to Europe to treat ague, or the malaria fever. Notable historical malaria cases I put down here. Alexander the Great died of malaria. George Washington had repeated attacks of malaria in Virginia. David Livingstone, the explorer, died of malaria in Zambia. And our favorite film star, George Clooney, also had repeated attacks when he visited Sudan and used his illness to highlight the severity of the disease. And there have been many other Western famous people, Vasco da Gama, Lord Byron, several popes and Roman emperors who are thought to have died of the disease. And why is that? It's really because malaria was common in Europe and the United States until the mid-1900s. This is an image here you can see of England. And we have the Fens, which are these kind of marshy, swampy areas around about London as well. And this is the number of malaria cases which are in these particular areas in the 1860s. And then also in the United States too, this is a map showing the southern states and up to the Great Lakes too. And it shows you the proportion of deaths from malaria compared to the deaths from all cases in 1870, and quite a large number of malaria cases there. In fact, we still get about 2,000 malaria cases each year, and these are from returning travelers who either return to their home countries for vacation or for work, and they bring back their parasites with them from these endemic countries. This is a map from 2018. You can see of the United States, with each of the states numbered according to the number of malaria cases. You can see New York City, where, of course, I've just come from, being one of the highest, but also California, too. These tend to be the ports of entry for travelers. And where do these malaria cases come from? Well, this is a map generated by the CDC indicating where the majority of the malaria cases picked up their malaria, and in particular West Africa, Central Africa, and East Africa as well, where Plasmodium falciparum gets picked up. And of course, some of you may have remembered a couple of years ago that there were 10 locally acquired malaria cases. These were the first in 20 years. We had in 2022 a couple of cases in Virginia. These are some of the announcements here. The CDC, the Morbidity and Mortality Weekly Report, excellent publication from the CDC, first announced these cases in Florida and Texas, closely followed by Arkansas, and then a bit closer to home, Maryland. There were 7 cases, in fact, Vivax cases in Florida, and 1 falciparum case in Maryland, which is a bit more rare. We partnered because we were asked by Maryland Department of Health to sequence the genome of these parasites from the Maryland case. So we got the blood, extracted the DNA, sequenced the genome, and you can see here when we compare to all of these other genomes that these actually came with another case as well. These cases here are most likely of Central African origin. And at the same time, Abai Tripathi at the parasite core was able to put the blood from the patient into culture and then transmit through mosquitoes. This is NF54, which is a common strain used to infect mosquitoes. And this is our MDH2 locally acquired strain of malaria. And you can see the number of oocysts. So these are the infectious stage on the midgut of the mosquito. And this particular Maryland strain also produced a large number of infections. So it's transmissible through the mosquito. So that gives you a little flavor then historically. What about the current status? Well, malaria in 2024, as Dean Ellen said, almost 263 million cases were accounted for in 2023, and then just over 500,000 deaths as well. So 83 malaria endemic countries, which is about 40% of the countries in the world. We have 44 countries which are malaria-free, and these do keep getting added to. Egypt and Cabo Verde and Belize were among the most recent countries to be made malaria-free. This is a map of the malaria risk. You can see primarily it is countries in Africa where people are most at risk, but also in South America, parts of India, Southeast Asia, and Melanesia as well. In fact, the malaria map is shrinking, and it has been shrinking for some time. This is a map from The Economist from a few years ago, and you can see that since the 1970s through to 1990, and then over the past 10 years or so, the malaria really has shrunk, in particular away from South America and then also from India. And in fact, my work in India, where I've based most of my career, really does show this. There's been a precipitous decline in malaria over the past 30 years. The figures are quite astounding because India has one of the largest populations of any country, about 1.4 billion. It used to have 23 million cases per year in the early 2000s, and now this has plummeted to 2 million cases, so real decline of about 80%. You can also see this visually on this map. This shows you India and the API. The API is the annual parasite index, and this is a measure of morbidity per 1,000 people. You can see how it changes from 2000 to 2015 to 2022. The API, which is really high, which is in red, is indicated, and the malaria has shrunk in particular to these 3 states, Chhattisgarh, Odisha, and Jharkhand, which is actually where we've been working for the past several years. These maps also show how malaria in India has decreased over the past 20 years. And some time ago, India brought out a national framework for malaria elimination and a national strategic plan as well, which was moving towards elimination of malaria by 2030. Although I don't think they will quite make it, they're actually pretty close. So malaria is a humanitarian issue. It's a disease of impoverished people living in resource-limited settings. These are some images from our field sites in India. This is a panoramic view of one of the villages that I'll be discussing a little later, where we've been doing a survey of some of the malaria camps. And you can see that there's no electricity, there's no running water. It's primarily an agricultural existence. And the housing structures as well are pretty basic, such that there's no protection from mosquitoes flying in here. So at the Johns Hopkins Malaria Research Institute, I actually thought that I would put a few slides up about the Institute for those of you who don't know what we do. We are innovating strategies that prevent and cure malaria. We've been continuously funded through Bloomberg Philanthropies since 2001, when the first gift was given. And it supports 16 faculty groups which are undertaking high-impact research on all facets of malaria. We are basic scientists, epidemiologists, immunologists, genomicists, clinicians, neuroscientists. And so we can bring the full force of all of our disciplines to this major global health problem. This is a list of all of the faculty here. I should also point out the previous director, Peter Agre, and Marcelo Jacobs-Lorena and Clive Schiff, who are emeritus professors. So what are we doing at the Institute? I've got a few slides here just to describe some of the projects that the different faculty are doing. So a big push is towards developing better methods for controlling mosquitoes. If you can control the mosquito, then you might be able to prevent malaria infections. So George Demopoulos and other colleagues are trying to develop genetically modified Anopheles mosquitoes, which will then be unable to transmit malaria parasites. Marcelo Jacobs-Lorena has been infecting mosquitoes with natural bacteria to prevent them from transmitting the parasite. And Conor McMenamum has been identifying human scent chemicals and how the mosquito detects them to develop innovative repellents and attractants. We're also developing new diagnostic tests and therapeutics. A group of faculty recently started to look into repurposing a known drug as a chemical vaccine, led by Terry Shapiro here. And a chemical vaccine then is one which is injectable and has slow release, long-acting, and provides weeks of protection, unlike a drug, which is very fast-acting. Dr. Matt Ippolito has been studying drug reformulation and selective deployment of different treatment strategies in Zambia. And my group, and I'll be talking about this a bit later, have been trying to identify new biomarkers to develop better rapid diagnostic tests. Faculty also are developing the next generation of vaccines. There are 2 vaccines which are currently approved for use in children in African countries, but they are not that effective. And so we are developing the next generation of vaccines using different technologies. Fidel Zavala, for example, is identifying human monoclonal antibodies that prevent infections. Prakash Srinivasan is developing a vaccine that will transcend the multiple genetic strains which are circulating around the world. And Fatini Sinis studies how many parasites it takes to develop a malaria infection, which is very important when you're assessing infection likelihood. I wanted to play this video. mosquitoes can creep people out, but we have these fantastic insectaries, about 7 of them altogether, looked after by Conor McMenamin. You can see that was a blood feed that was happening. So we feed the mosquitoes on human blood, which is generated by Terry Shapiro. And we can do all sorts of experiments with them. For example, making GFP fluorescent proteins to show how the parasite, oops, shouldn't have done that. Oh, I'll have to start again, which will show how the parasite is moving through the mosquito. We produce, I think it's 60,000 mosquitoes a week. Our mosquitoes are sought after by other labs, and so we routinely ship them to other places, other laboratories. And it enables us to really work towards completing the life cycle in the laboratory, as you can see here. Okay. The Institute serves both the public and the scientific communities with conferences. We have 3 conferences each year, so we are busy. And at the moment, because it is World Malaria Day on Friday, we are all busily planning a World Malaria Day conference organized by our mosquito vector biologists, building better mosquitoes. So that will be happening here at the Bloomberg School of Public Health to mark World Malaria Day. But we also have Vector Encounter as well, where all of the, or many of the people working on vectors will come and discuss on a whiteboard the research that they're undertaking. And we get all of the young junior malariologists together once a year as well, usually just before a big conference. And this is for the future of malaria research. And last year we held it with the University of Maryland, which was also very successful. And we have the Malaria Minute here, which is a podcast which is shown twice a month, where investigators who have published a recent paper can talk about their work. So JHMRI scientists also partner with researchers at field sites in Africa and Asia. We don't just work on our research in a vacuum. It has to then go out into the field to be tested. We support Matcha Research Trust in Zambia through a infrastructure grant, and this provides housing and lab and insectaries and this amazing 5,000 square foot mosquito sphere that was developed. And this is just in Matcha, which is just in the southern part of Zambia. We also have the NIH International Centers of Excellence for Malaria Research, which I'm going to talk a little bit more. In fact, we have 2 of those. Bill Moss leads the Southern African isomer here. The Indian isomer actually is led by people at the London School of Hygiene and Tropical Medicine, but we still have 1 foot in the door as well. And we have other field sites too, where we do a lot of our research, for example, in Ethiopia, Kenya, and Uganda. The NIH-funded isomer program has been going on since 2010. It's really a phenomenal program, and actually I think quite visionary. It combines research with capacity building, training, transfer of technology to endemic sites. There were 10 centers that were funded by NIH in 2010, and you can see the regions that they covered here. In 2024, 2 renewals later, you can see that has shrunk to take in consideration where malaria now is most prevalent, which is in African countries, but then also Southeast Asia too. And I'm going to, let's see if I play this. There we go. So these centers then form a network of interacting regional centers across the globe. They're cooperative regional centers funded for 7 years, which is quite unprecedented in NIH terms. Actually, this has just been cut back to 5 years, but it was 7 years in the past. And we all have to do epidemiology studies. Obviously, we need to see what malaria looks like in the regions where we operate. And we undertake human vector and parasite studies. We have to be responsive to change. If the epidemiology changes, our research needs to change. And as I mentioned, there's this focus on capacity building and transfer of technology. We're not just flying into an area doing what I call colonial science, collecting samples, flying back out again. We're really working with the local people, the scientists, and the researchers together. And there is this direct relationship between the isomers and in-country control programs so that our research then goes on to be implemented in the field. And this movie, which was taken by Bill, shows you some of what the field teams get up to. This was taken in Zambia, and this woman has her finger pricked and blood is being collected on these cards. And also she will have been tested for malaria too. Looks like her little child's not very happy there. So I've given you an idea then about the isomers. I've given you an idea about the Malaria Research Institute. I'm going to talk a little bit about my research. And I'm going to do that in the context of an India isomer project that we did and the reasoning behind it. So in 2017, there was a novel elimination tool called these Daman malaria camps, which were implemented in Odisha state in India. This is Odisha state here, this is 2016, and you can see this annual parasite index again is incredibly high, greater than 10 in many of these red areas here. And so malaria in Odisha was really thought to be enormous, with less than 4% of the population, more than 40% of the malaria cases occurred in this particular state. And so the government of Odisha implemented what's called Daman, which stands for Malaria Control in Inaccessible Areas, in 2017. And the Daman initiative was delivered through these malaria camps in remote villages across the state, 18 of 30 districts in total. And the procedure was quite clear. They started off with mass screening and treatment, or MSAT. There were also vector control measures, so long-lasting insecticide nets, indoor residual spraying, but then also community mobilization, training, and participation too. So this is what a malaria camp would look like. First of all, the Odisha government field teams would have to get to these inaccessible villages. You can see these are some of our team members that were following along with the Odisha government through this quite rugged terrain, having to wade through streams, also issues during flooding of getting vehicles through there. And the camps would occur when all of the villagers were asked to congregate in the center of the village, and there would be mass malaria screening. So everybody would get an RDT, a rapid diagnostic test. There would be health checks of children under the age of 5 and of pregnant and lactating mothers. And then there would be a first round of insecticide spraying internally, followed by long-lasting net distribution, and then what's called an intensified routine program. And then there will be a second camp after the monsoon, where in fact there should be fever screening here. Fever screening, but then basically repeating what had been done previously, and then the third round as well. So very intensive groups of field workers going to very remote villages and implementing all of these different control measures. And malaria in Odisha state decreased enormously. Here's 2017, shown in red, the cases of malaria, 2018 in green, and then you have malaria cases in 2019. And the World Health Organization lauded these particular malaria camps, as you can see here. WHO pat on Odisha's sting operation, it says. So we, through our International Centre of Excellence funded by NIH, evaluated actually if this Daman malaria camps really did reduce malaria prevalence, or was the malaria just decreasing in the state anyway. We did this through a trial. We identified 15 villages shown here, and these villages contained about 2,500 people, and they were enrolled into different arms. And the different arms were as followed. They had a malaria camp, or it was a delayed malaria camp, or they'd already received malaria camps. And you can see the intervention timeline along here. We started in August 2019 and were able to do our baseline. But then soon after that, of course, the COVID-19 pandemic hit. And so it was a challenge really to do many of the follow-ups until we got to follow-up three indicated here. So what did we find? We found that, yes, indeed, intent to treat analyses suggested the Daman malaria camps were associated with lower malaria incidence. So for every person who was enrolled in the study, they would have had a rapid test taken and then also a PCR test as well. And you can see at baseline how many of the people in different arms had plasmodium infections, and then subsequently at follow-up 3, how many of them did. So it really seemed that these Daman, very focused malaria camps seemed to be associated with lower malaria, and this was published a couple of years ago. But one of the big findings for us was that how this effectiveness trial basically identified a high RDT failure and a high percentage of what's called PF-HRP2 gene deletions. So let me back up a little and tell you how malaria is diagnosed. It's diagnosed through the use of these rapid diagnostic tests. You can see this is a nitrocellulose filter paper here, and a drop of blood from a finger prick is posted on one of these filter rapid tests, and antibodies then are used to identify antibodies in the patient which have been generated because of these particular parasite proteins. And the parasite proteins which are used routinely are what's called histidine-rich protein 2, HRP2, and then also LDH. This is really for a different species. And the problem is, about 10 years ago, that the HRP2 gene in the malaria parasite was found to be deleted, and strains of this have been spreading globally. So our work showed then that we had not only a high number of RDTs which said they were negative, whereas in fact the patients had malaria parasites, but there was a high percentage of PF-HRP2 gene deletions as well, and that's really shown here. So 80% of our infections were not detected by RDT, 80%, that's pretty phenomenal. And of the RDT negative infections, about 62% had this PF-HRP2 gene deletion. This was actually quite similar to previous studies in Odisha, which showed about 60% HRP2 gene deletions. And so, of course, the high RDT failure rate, especially as RDTs are used in these Daman camps to identify malaria cases, and the spread of this HRP2 deletion is very concerning, and it does threaten the road to elimination, those roadmaps in India that I was showing you. So what's the solution? Well, part of the solution, of course, is identify better falciparum biomarkers to develop better rapid diagnostic tests. And we have started to do this in the past couple of years in my lab, first of all by looking at puffs. So what are puffs? Puffs are proteins of unknown function. So let me back up a little bit here again too. I was involved and led the projects to sequence the very first malaria parasite genomes, first Plasmodium falciparum, and then Plasmodium vivax in the early 2000s. And of course, this was fantastic because it identified all of the genes and the proteins in the malaria parasites for the first time and really expanded the basic biology and our knowledge of the parasite metabolic pathways, what was found on the surface. They were very informative. However, as you can see here, a number of the proteins just had no function. they were what we call hypothetical. We identified the gene, we knew that they were expressed, and there was probably a protein. We had no idea what it was. It's really shown in this table here. So the falciparum proteome, the total predicted proteins, 5,200, so that means there are about 5,200 genes. And look how many are hypothetical. We have no idea what they do, 60%. And that's very challenging because it means that if we want to, we're working in the dark, not knowing what these genes and proteins do. Another way to view it here is this is one particular chromosome, chromosome 1. These are all the genes shown as diamonds along the top. And each of these genes can be given what's called a gene ontology description, which shows basically where it's located or what its function is. And you can see how many of them are white. That's because we have no idea what any of those Go terms are. So Harsh Srivastava, who's a PhD student in my lab, turned to machine learning. And so let me tell you a little bit about machine learning. So it's a specific method within AI that uses algorithms trained on data to enable machines to learn and make predictions. You may have heard of it more recently because 3 members who got the Nobel Prize in Chemistry had also really developed and pioneered these particular ML techniques for AlphaFold. So AlphaFold is an ML model. It can predict protein structures. Previously, prediction of protein structures would take years if it was successful at all through generating crystals. AlphaFold can do that in now a matter of hours. Protein language models are actually a type of machine learning that learns how to represent protein sequences. And PLMs, protein language models, can be used in a number of different areas. You can see here function prediction, which is what we have used it for, but also mutation prediction, protein structure prediction, and then protein design, which I'll also be talking about in a minute. So P. falciparum puffs cannot be annotated using existing sequence-based techniques, but PLM can come to the rescue. And Harsh has developed a model, Plasmodium Function Predictor, PFP, that relies on these structure-function relationships to predict protein functions specifically for Plasmodium. And PFP models predict protein Go terms, the molecular function, biological process, cellular component, as I showed here. And it actually outperforms many of the other ML programs, such as DeepGo, which have been used to do this. So what does it mean for the falciparum genome? It means that we have increased, or I should say reduced the number of hypotheticals by 16%, increased the full annotations by 15%. So all in all, an increase in what we know about the genome now. These falciparum puffs are actually part of many biological processes. It's not as though there's one metabolic pathway in falciparum that has no homology to anything else. They are really distributed throughout the metabolism. So that is great. We're now Harsh is developing this model to run it on Plasmodium vivax, which actually is in a worse state than Plasmodium falciparum, and then some of the other human malarias as well. And this was done in collaboration with Rich Bonneau, I should say, and colleagues here. So that's one angle we're taking, the hypothetical genes that we weren't able to identify during the genome projects. Another approach is being taken by PhD student, Maria Nikolkova, shown here. And we have one of the richest data sets generated from a visit that she and I both took in 2002 to Ethiopia, working with Delina Soyulaha and colleagues here. And that is usually when you take a sample from a patient, that sample the genome will be sequenced, or you'll sequence the transcriptome. But in fact, we generated all of these omic data sets to generate a multi-omic data set. The genome, transcriptome, metabolome, lipidome, proteome from every single sample. And that's a very rich data set to analyze. Maria trained the team in Ethiopia, who then went out to the 3 healthcare clinics. You can see that they collected vacuotainers, so these were blood taken directly from veins for all of these different types of molecules, RNA and DNA and so on and so forth. But of course, the fact is that we had to test many of these subjects too for their plasmodium infection status. We may have got 75 RDT positive and 75 RDT negative, but we already knew that the RDT negatives could be false negatives. And sure enough, it turned out we only had about 40 true plasmodium negative samples. Out of our falciparum samples, we had about 30, and 23 of those were RDT negative, shown here. And we took these 23 rapid test negative, falciparum positive, and Maria had a look to see if there were HRP2 and 3 gene deletions. And sure enough, the majority of them did have these deletions. And then because we had the transcriptome, we had all of the transcripts, we could then have a look to see whether there were any transcripts, and the HRP2 positive there were, and the HRP2 negative there weren't. So what have we done with this very rich multi-omics data set? Well, we started to look at LC-MS proteomics of the plasma from the falciparum subjects to identify new abundant protein biomarkers. It's quite interesting that the PF-HRP2 protein, which all the rapid diagnostic tests in the world use, many of them, I would say about 90% of them, the way that it was developed into a rapid diagnostic test is a little unclear. It's definitely one of the more abundant proteins in plasma, but we hypothesize that there'll be far better proteins to try and identify. So we have taken, and this was work done by Walter and Harsh as well, 8 of these Ethiopian subject plasma samples, all of which were PCR positive, some of which were RDT negative, some which were RDT positive, and run them through LC-MS to identify what are the most abundant proteins, malaria parasite proteins, in the plasma. We identified about 1,000 altogether, 61 abundant proteins present in at least 5 out of 8 isolates. This upset plot at the bottom here is just basically a giant Venn diagram, and it shows you quite clearly that, in fact, there isn't a significant amount of overlap. So, in fact, we do need to run more of these samples, the plasma samples, from more patients through proteomics. So that this number of abundant proteins will probably change. But this is a list of the abundant proteins. 61 in 5 out of 8 isolates. This is not very useful, I'm afraid, but it just shows you the UniProt ID, which samples, which patients they were found in, and their function associated here. But of course, really what we want to do when we narrow down better biomarkers is we want to identify proteins whose genes show the least number of genetic variants. There's no point in designing a rapid diagnostic test based on a biomarker if that's going to be really variable in different African countries. So that's one thing that we're starting to look at. We also want to make sure the protein stability is good enough to work this up into an RDT. And we want to make sure that the proteins are coded by genes that are essential, so they're not deleted, like the HRP2 gene has been deleted in all of these strains. And one way of identifying those is by looking at the core regions of falciparum chromosomes. So those contain genes which are less likely to be deleted. We'll also cross-check to see if any are found in the exportome. It means they're more likely to be exported out of the infected red blood cell. And also check against the known malaria gene atlas, which contains gene expression data to make sure that the proteins are expressed in all these different stages. Finally, once we've done that, and we're continuing experiments as I speak, we will once again use protein language models. And this will be to design de novo protein binders to these new biomarkers. Protein binders then are proteins designed to bind to Plasmodium proteins. They could be antibodies or mini proteins or scaffolds. And we can use protein language models to design these. And then we will put these on nitrocellulose strips and then take them to Ethiopia or to India and to test them out. Okay. So that's a little bit about how omics and machine learning, the sort of techniques we're using in my lab here. I did want to end on the implications of the cuts to the US global health programs. I know this is sort of on everybody's mind, and I wanted to tell you about what this means for malaria in the lead up to World Malaria Day. So the US is the top donor government to malaria efforts, and primarily through two initiatives. The first is the President's Malaria Initiative, PMI, but then the second is the Global Fund to Fight AIDS, TB, and malaria. I'm going to be talking about PMI in particular. PMI was founded in 2005, and the Congress created US Global Malaria Coordinator, who was recently appointed a couple of years ago, Dr. David Walton, as appointed by the president. Of course, he no longer has this position because since USAID was cut, so was PMI. This is the annual report that PMI sent to Congress in 2024. And this is one of the figures from it that really shows you the impact that they have had in about 30 or 40 African countries, and then also in the greater Mekong subregion. So if you just bear with me, you can see the year when they started operating up until 2023 on the top, and then on the bottom you've got the countries that PMI started to work in, and then this is the dollar value increasing how much was provided each year. And what was that for? This is also from the report. PMI delivered, and this is just last year, 37 million mosquito nets, 15 million preventive treatments for pregnancy, 48 million seasonal preventive treatments, 103 million rapid diagnostic tests, 63 million malaria medicines. And so they're thought to have benefited at least 700 million people, and also then to have caused the decline in malaria, as stated here with these numbers, decline in malaria death rates of close to 50%. So what does this mean then? We have the funding freeze, the stop work orders, the dissolution of USAID, the cancelled awards, the reorganization. Well, several different institutes have done analyses to see how this might impact malaria over the next few years. You can see here there was a USAID memo, and it shows that, and it indicated, this was an internal memo, that up to 18 million cases and 166,000 deaths are going to occur in addition if PMI is halted permanently. And these were similar numbers that the Malaria Atlas Project did a geospatial modeling. They came up with similar numbers to that too. The WHO did a rapid survey in March, and it showed that of the 108 country offices, 50% reported moderate to severe disruptions to malaria services, and that's medicines, rapid diagnostic tests, so on and so forth. And Rollback Malaria has this very useful dashboard showing supply chains, and they have indicated that there are only 6 countries with a 3-month supply of RDTs and 5 countries with only a 3-month supply of ACT. It's quite visible on their dashboard. So unfortunately, it's all very depressing. I also talked to some of our collaborators in African countries. So Dawit Hawaria is our main collaborator in Hawassa in Ethiopia. I know this is a little text heavy, so apologies. But this is what he said. The PMI office is already closed. The 2 highest malaria burden regions, Gambela and Benshangul Gumuz, which were entirely supported by PMI, the office is now shut down, so that's disrupted the supply chain. The access to bed nets now unavailable, and the National Malaria Programme intended to investigate malaria cases in the capital, Addis Ababa, and that's no longer going to happen. And another colleague, Melissa Conrad here, who sent me a very personal and emotional email about this when she was in Uganda, where she works at some field sites, about how the Central Public Laboratory was told not to come back to work, how it's unclear who's going to pay for electricity, internet, and water. And this was right when the Ebola outbreak started too. And she finally signs off, I've been relatively calm so far, but I'm starting to realize any optimism is probably displaced. So it's a sorry scenario, I'm afraid. Okay, I'm going to leave it there. I'm sorry it's not a very uplifting ending, but these are acknowledgements that I'd like to leave you with. In particular, I want to have a big shout out to my lab, because many of them that you can see here and listed here came from New York University. They put enough trust in me to follow me down here, and that takes a lot. So a very big thank you to all of you. And then also our partners in India, shown here, and our partners in Ethiopia, especially Delina Soyulaha, and of course the people of India and the people of Ethiopia, because we are just visitors in their beautiful countries. So thank you.
Thanks so very much, Shane. That was it was incredible, and it was great to start with the history and move forward and look towards the future. Ending on not such a positive note, but I'd love to ask you to start the questions. Where's the hope, and how can we move forward given the current environment?
Yeah.
Not an easy question, I know.
Yes, no, that's very true. Well, I think the hope is in science, right? The hope is in research. There are definitely new initiatives, new tools which are coming to the forefront, some of which I mentioned, and several of which we're developing here at the Malaria Research Institute. So I think that is good. I do know the World Health Organization has got together with other countries to provide additional funding and support for those countries who have lost support through PMI. So that's one positive thing too. Yeah.
Questions, comments? Yeah, go ahead. And if you have a question online, either raise your hand or you can type it in the chat and we'll call on you.
Yeah, thanks, Jane. Very interesting and, as you said at the end, a bit depressing talk about where we are in the scheme of things right now. Steve Harvey with International Health. I've done a lot of work on RDTs, and I'm curious in your thinking about RDTs if you have had put any if you have any developments on samples other than blood for detecting antigens. It'd be so much nicer if we could work at the community level without having to deal with lancets and sharps boxes and all the things that are associated with needing blood as a sample, and could use instead saliva or urine or stool or just about anything other than blood. Any developments there or potential developments there? Thanks.
But not really, but through colleagues actually at the Malaria Research Institute. So there are people who are developing potential breath rapid diagnostic tests. So studies have shown that infected people give off breath which is different compared to uninfected people. And so there's research in that area. Connor McMenamin here at the Institute is somebody that you should talk to about this as well, who has been identifying the scent given off by infected people, which could then be developed into attractants for mosquitoes. So you pull the mosquitoes away from people. That's another way. That's not a diagnostic, but so there are other definitely other platforms out there. I would say, however, though, that I feel as though blood if you have parasites in your blood, you have the parasite. So to me, it's the go-to molecule of choice. That's what we're going after, and it's just a little finger prick.
I'm not squeamish about blood, but disposal of lancets and things like that is tricky, particularly when you're doing it at the village level where there may not be good biosecurity measures.
Right. And also children, actually. Children have such tiny little fingers.
They're not so
Or,
those little fingerprints.
or toes.
Other questions?
I'm interested in your protein function.
Wait a minute, hold on. Just let's get, sorry, so everybody can hear.
I'd like to learn more about how to predict the functions for unknown proteins and how far you've gotten or how it's been applied to other microorganisms.
So I'm not sure, actually. I think we're the first to have applied it. So what a lot of the machine learning models have done is to learn on organisms which are not closely related to Plasmodium parasites. So one thing that we did was take the SAR family, which is the big phylum, the big grouping, which contains Plasmodium and other parasites too. And we took all of the proteins, all of the genomes from that to train our models. And that's not been done before, actually. That's the first.
Yeah, Peter.
Well done, Jane. I have one question that I've never really gotten a clear sight of. The Peruvian Indians had the cinchona bark to treat malaria, but there was no malaria. What were they treating?
Oh, interesting. I hadn't thought of that. Well, was there no malaria in 1638? I mean, it was brought over by the slave trade, and I think that started 1500s, 1600s, 1700s. So it may have just started to be there. Maybe? Do you know the answer to that? Because no.
They were, the observation was it would stop shivering, working in the mines, or it was just an observation. It wasn't, I don't know if they really equated treat malaria with taking the bark. And interesting around the Pope, 1623, the Pope died of malaria, and also being closed. The Jesuits, then through the Catholic Church, were looking for, well, the Pope had, put a call out for malaria, and the Jesuits in Peru said, well, we have this bark that can stop the shivering and chills, and then they brought it, and then the Jesuits played a hand. I don't know if they were the leading hand, but played a hand in associating. They still didn't have the diagnosis back then, 1600, so they were just associating periodic fevers with taking the bark. I mean, Laveran didn't do the blood film, and we didn't really, diagnose until the late 1880s, 1890s or so.
Okay.
Yeah, it was. We had the cure before we knew the etiological agent.
Interesting. Thank you.
There's a, David, to follow up on your comment and Peter's question. There's a great book, I'll look up the title of it in a minute, that traces the history of cinchona bark. And the Jesuits had a monopoly on it for a while and made quite a bit of money at it. And it was of great interest to the Catholic Church because about a third of the College of Cardinals would die of malaria every time they'd come for a conclave to elect a new pope. And of course, as most of you probably know, the name of the disease comes from the Latin malare, bad air, which was what you drove through in your coach on the way into Rome in the marshes on the outskirts. And cinchona is what could protect you from that or cure you after you got sick.
We've got a couple of historians. It's wonderful.
It's pretty fascinating history.
Yeah. Any other? Yeah, go ahead. Oh, wait a minute.
I can speak.
They can't hear you online. Sure.
Yes, thank you very much for this comprehensive tour of so many facets and the in-depth tour of the proteins. My question is, how much of the transmission in your sites comes from Stephen's eye, and whether this complicates but also maybe opens avenues for control?
So Anopheles stevensii, really interesting species of mosquito. So in India, Anopheles stevensii is found primarily in the south of the country. There are 6 different vectors in India, at least 6, that transmit malaria parasites. And stevensii is found more in the south, which is where Plasmodium vivax is found. It's also associated with urban areas too. But the really interesting thing, maybe you're alluding to this, is that it has invaded countries in the Horn of Africa recently, and that's real huge, enormous cause for concern. So it seems to have come over from Asia into African countries, and there are surveillance teams out there trying to track to see whether that's going to increase the amount of urban malaria. Which is why seeing those cuts to the PMI initiative to look at malaria cases in Addis Ababa, the largest population, largest city in Ethiopia, is quite concerning. So yeah, very interesting species that.
I think we have time for 1 or 2 more questions. If nothing online, Lataska? Okay. All right. Well, thank you again, Jane. Please join me in thanking Dr. Carlton for the.
Thank you.